Hello everyone, in this video I'll be explaining on the topic immunity, lymphoid organs and tissues. Coming to the classification of immunity, immunity is classified mainly into two types, innate immunity and acquired immunity. Innate immunity is defined as the resistance offered by the host against infection caused by the microorganisms present from the birth, which is due to genetic or constitutional makeup of the host. Innate immunity is broadly classified into three types: species immunity, racial immunity, and individual immunity. Species immunity is the resistance against infection which is present in the members of species. Example, frogs are resistant to anthrax at 20-25 degrees Celsius but susceptible at 35 degrees Celsius. Racial immunity is the resistance against infection present in different races within the same species. Example, American black are more susceptible to TB than the white. Third is individual immunity. It is the resistance present in different individuals. Example, homozygous twins have identical resistance of susceptibility to infection, which is not seen in heterozygous twins. Next is acquired immunity, which is also called as specific immunity or adaptive immunity. It is classified into active immunity and passive immunity. Active immunity is the immunity or resistance acquired in the individual against an infection after the contact with an antigen or when after they encounter an infection or a disease. And this active immunity is classified into natural active immunity and artificial active immunity. Natural active immunity is defined as the resistance developed in an individual as a result of clinical or subclinical infection. Example, infection with measles, mumps, or chickenpox. Second is artificial active immunity. It is defined as the resistance developed in an individual following the administration of vaccines. Example, oral polio vaccines. Vaccines are the preparations of consisting of live or kill suspension of microorganisms or the products that is used to induce immune response in a host. Next is passive immunity, which is defined as the resistance achieved by the introduction of preformed antibodies. Passive immunity is again classified into natural passive immunity and artificial passive immunity. Natural passive immunity is defined as the resistance that is transferred passively from the mother to the fetus or baby. Example, neonatal tetanus can be prevented by active immunization of pregnant women. Next is artificial passive immunity, which is the resistance that is transferred to the individual by the administration of antibodies. Next is the lymphoid organs and tissues. The lymphoid organs are, the first is the lymph node. It is the principal lymphoid organs in the body are the lymph node. Lymph node have two basic functions, both concerned with body protection. One is as lymph node is transported back to the bloodstream, the lymph node acts as lymph filter. Macrophages in the lymph node remove and destroy microorganisms and other debris that enters the lymph from the loose connective tissues, effectively preventing them from being delivered to the blood and spreading to other parts of the body. Second function is they help activate the immune system. Lymph node and other lymphoid organs are strategically located sites where lymphocytes encounter antigens and are activated to mount an attack against them. A lymph node has two histologically distinct regions, the cortex and the medulla. The superficial part of the cortex contains densely packed follicles, many with germinal center, heavy with dividing B cells. Dendritic cells nearly encapsulated the follicles and about the deeper part of the cortex, which primarily houses T cells in transit. The T cells circulate continuously between the lymph nodes, blood, and lymph, performing their surveillance role. Next is the spleen, which provides a site for lymphocyte proliferation and the immune surveillance and response. But perhaps even more important are its blood cleansing function. Besides extracting aged and defective blood cells and platelets from the blood, its macrophages remove debris and foreign matter from blood flowing through its sinuses. The spleen also performs three additional or related functions. That is, first, it stores some of the breakdown products of red blood cells for later reuse. Example, it salvages iron for making hemoglobin, and then it releases others to the blood for processing by the liver. Second function is it stores blood platelets. Third is it is thought to be a site of erythrocyte production in the fetus, a capillary that normally ceases after birth. Like lymph node, the spleen is surrounded by a fibrous capsule, has trabeculæ that extend inward and contain both lymphocyte and macrophages. Next is coming to the bone marrow. Bone marrow is a sponge-like tissue situated inside the bones. Most defense cells are produced and then also multiply here. They then migrate from the bone marrow into the bloodstream and reach other organs and tissues where the defense cells mature and specialize. At birth, many bones contain red bone marrow, which actively build defense cells. During the course of life, more and more red bone marrow turns into fat tissues. Adults only have red bone marrow in a few bones, for example, in the ribs, in the breastbone, and in the pelvic bone. Next is the thymus. The bilobed thymus has important functions primarily during the early years of life. The thymus is the site where T lymphocyte precursors mature to become incompetent lymphocytes. In other words, the thymus is where T lymphocytes become able to defend us against specific pathogens in the immune response. Recent evidence suggests that Hassel's corpuscles are involved in the development of a class of T lymphocytes called regulatory T cells, which are important for preventing autoimmune response. In addition to its lack of follicles, the thymus differs from other lymphoid organs in two other important ways. First, it functions strictly in maturation of T lymphocyte precursor, and thus it is the only lymphocyte or lymphoid organs that does not directly fight against. In fact, so that so-called blood thymus barrier keeps blood-borne antigens from leaking into the cortical region to prevent premature activation of the immune lymphocytes. Second, the stroma of the thymus consists of epithelial cells rather than reticular fibers. These epithelial cells provide the physical and chemical environment in which T lymphocytes can become immunocompetent cells. Next is the thymus, the tonsils. Here, the tonsils are the simplest lymphoid organs. The crypts trap bacteria and particulate matter, and the bacteria work their way through the mucosal epithelium into the lymphoid tissue, where most are destroyed. It seems a bit dangerous to invite an infection this way, but this strategy produces a wide variety of immune cells that have a memory for the trapped pathogens. In other words, the body takes calculated risks, high risks, early on during childhood for the benefits of heightened immunity and better health later. Next is Peyer's patches. Peyer's patches or aggregated lymphoid nodules are large clusters of lymphoid follicles structurally similar to the tonsils. They are located in the wall of this distal portion of the small intestine. Lymphoid follicles are also heavily concentrated in the wall of the appendix, a tubular offshoot of the first part of the large intestine. Peyer's patches and the appendix are in ideal position to destroy bacteria which are present in large numbers in the intestine before these pathogens can breach the intestinal wall, and also to generate many memory lymphocytes for long-term immunity. Peyer's patches and the appendix and the tonsils all located in the digestive tract, and the lymphoid follicles in the wall of the bronchi, organs of the respiratory tract, and